Thank you so very much. I have to admit that I had an invitation last year to come, and I said, what the heck am I supposed to talk about? You know, I do systems biology, talk about nano and tele, and so this year I got another invitation. I said, these people are insistent, and I just do it. If they think it fits, okay, I'll do it. So ignore the abstract in the book, but I will talk about the title that is given there. And let me see. Okay. So I want to talk about systems modeling and systems biology, and since this is sort of an outside topic in this conference, I thought instead of giving a very technical talk, I give an overview of some of the issues that I see that where systems biology could help in predictive medicine and maybe overlap with some of the things that most of you are doing. So I want to talk about the grand challenges of systems biology and then talk about the drug pipeline. And most of you have probably seen the drug pipeline and the different phases of that. And I just want to focus on where systems modeling, biological systems modeling, might play a role in the drug pipeline and maybe help move the screening earlier in the drug pipeline. And then I will talk about pathway modeling and a little bit about predictive health. And let me give you a caveat. So everything will sort of sound like hand waving, but there is some real science behind it, and it will not necessarily show up. And I thought I should say that. And if you're interested in these things, you know, I'll be happy to talk with you or send you papers or whatever. But don't be fooled by, you know, looking at pretty pictures. So the grand challenge of computational systems biology is that we have cells, or we have a tissue, or we have an organism. And what we would like to do is convert that into diagrams, and with, you know, arrows and boxes and maybe different colors and different types of arrows. And we want to take those diagrams and convert them into mathematical equations or systems of equations. And in typical cases, these are differential equations, but not necessarily. There are other things. Then we need to convert that into computer code. And if we have done that, the main thing is we want to come back and gain insights or create some new hypotheses based on the insight that we have learned from the computation analysis. So why do we want to do that? So what are the rewards for solving these grand challenges? Well, of course, the testing would be incomparably faster and cheaper. We can explain root causes of observations. So if you see that phenomenologically you have some problem at the end of the system, you can go back and find out where exactly this problem comes from, or what combinations of problems have led ultimately to the observation we make. We would like to make reliable predictions, and clearly if you have a computational model, you can make hundreds of thousands, millions of predictions in a very, very short period of time, much, much faster than any organismal system or in vitro system. So we can explore whole parameter spaces with these types of models, and as long as the models are, or as soon as the models become reliable, we can make very interesting predictions of what these systems are doing. Then ultimately we want to create novel biological systems, and that's going into synthetic biology and maybe coming a little closer to what you are doing. And in a way we could reduce some animal experiments and ultimately improve medicine. So here's sort of I put this in yesterday after I'd heard the talks, and here are sort of the relationships to what we are doing, what you are doing, the way I see it. So we have some medical examination or methods of telemedicine. And from that we get some biomarker profiles. And here this is a mass spec, but it could be other things like blood pressure or pressure inside the heart or things we heard about yesterday. And that would lead to the identification of a problem and of the affected tissue. And then you could go into location issues, maybe with nanodots, and our department is doing a lot with that, with quantum dots and those things. And you can also go into DNA arrays and look at the genetic predisposition, the genetic makeup, and the expression, changes in expression. Those things all would go into a computational model. This model would allow us to personalize the system to a given patient, to a given person, and then we could customize a viral vector or a nanobot. I don't know much about nanobots. We heard yesterday about the potential of those. And then not just kill the cell that is affected, but maybe correct it in some way. And then everybody would be happy. So here's the drug discovery pipeline very quickly. You know it starts with the discovery phase, and then there's the preclinical development, and the clinical development, and then the postclinical. We have the animal studies, the clinical trials, and so on. And I will not go through this in detail, just say that it takes about 10 to 20 years, and it may cost about $1 billion to come up with a drug that actually makes it to market. And all these steps in that drug pipeline are necessary, and we do not want to cut corners. So the only thing we can really do that would help us in reducing the time and the costs involved with the drug development process is to screen out things earlier than we may screen them out now. So once a new chemical entity, an NCE, goes into clinical phase 1 or 2, it becomes very, very expensive. And if we could, with any sort of modeling or other methods, move the screening out earlier, it would save an enormous amount of effort. So there are different types of models, and here are some that are being used in the pharmaceutical industry right now, and I'm not going to talk about them. So there's screening, there are the quantitative structure-activity relationships, and there are binding predictions. And I will not talk about them because that is sort of going on right now, and I don't need to mention it. There are the PBPK models, that's physiologically based pharmacokinetic models, and they have been in the industry for some while, and they are becoming more interesting and more sophisticated. So essentially what you do is you divvy up the body into compartments, like the blood compartments, brain, lung, and so on. And they are connected, of course, for instance, through the bloodstream. And now you can put a drug into the bloodstream or into the stomach or into the lung and see the kinetics or the dynamics with which these drugs go into different compartments, how long they stay there, whether they come out unharmed in some sense or whether something happens to them. And the different compartments can respond very, very differently. For instance, if you have fatty tissue, it usually keeps lipophilic substances for much longer. If a compound goes into the liver, it is at risk at least of being metabolized, which may be desired or may not be desired. You create breakdown products, which you could follow in the same model. And the beauty of this is you can set up these models in a mouse experiment, and you can validate them. You get the parameters out, for instance, the diffusion rate, the retention time, and other parameters. And then to some degree, you can scale up these models by saying, well, a mouse weighs 20 grams. We heard a human is 3,500 times heavier.
3,500 times.
That is 3,500.
Oh, you are.
Sorry. I listened to you. So 3,500 times bigger. If we could look at the volume of the liver or volume of the fatty tissue, then we can make deductions, mathematical deductions of how long a particular drug would stay in a particular compartment. And these models are very successful, and I think they will become more successful when we learn more about the degradation processes in different organs and kinetics of excretion and so on. Here's something that I was not aware of until last year when I talked with somebody from the pharmaceutical industry. And he said a big problem where we use systems modeling is actually receptor binding. So the situation is that we have a disease process, and the disease process has something to do with ligand binding to a receptor. And very, very many diseases are of that nature.